second objective is to describe the communication across chemical synapses and really describe how neurotransmitters are released and what happens on the adjacent neuron after those neurotransmitters are released. Okay, so let's talk about the anatomy of a synapse. Okay, so we have our presynaptic neuron and our postsynaptic neuron. And again, this connection is happening usually in the the central nervous system, so the brain or spinal cord. So this is your first neuron, this is your second neuron. And this is the area that we're really focusing on here. There's a presence of a couple of structures we'll talk about. The first one is the voltage-gated calcium channels that are present at the axon terminal. And I'm using the words axon terminal and terminal bouton interchangeably. They're the same structure. This ending terminal of the axon here, that first axon. Okay, so there the presence of voltage-gated calcium channels. There are vesicles that have neurotransmitter in them, and we call these synaptic vesicles. So these tiny little vesicles here that have neurotransmitter in it are called synaptic vesicles. We also have the presence of some type of receptor on the postsynaptic neuron. And this receptor is going to be unique to binding whatever this neurotransmitter is. Okay. We also have enzymes. So this enzyme, we'll talk about exactly the name of this enzyme, but I want you to think of this enzyme as being responsible for degrading any excess neurotransmitter that's left at the cleft. And the cleft is this region. This is called the synaptic cleft. This space or this opening, this region of communication between these two neurons at your synaptic cleft. So any additional or excess neurotransmitter that's lingering in the synaptic cleft will be degraded by this enzyme. We'll talk more specifically about what the name of this enzyme is as that becomes important later on. We also have the presence of reuptake molecules. So this purple ball, which is present on the presynaptic neuron, is a reuptake molecule, and that is going to be responsible for reuptaking any of those degraded neurotransmitters that have been broken down by this enzyme. So this enzyme will break down those neurotransmitters into smaller pieces, and then they can be reuptaken into this terminal bouton, recycled, repackaged into a new vesicle to make some new synaptic vesicles, which have new neurotransmitters. Alrighty. So these are the important structures in understanding what takes place at a synapse. All right, I'll go over those again. We have a voltage-gated calcium channel specifically. We have our synaptic vesicles, which contain some type of neurotransmitter. We'll talk about what those are later. We have a receptor that is unique to this neurotransmitter on the postsynaptic neuron itself. And then we also have an enzyme and a reuptake molecule that are responsible for degrading and reuptaking the excess neurotransmitter that's left. Okay. Okay. So let's talk about the steps of synaptic communication. What's really happening for one neuron to signal to the other? Okay, synaptic communication. Okay, so synaptic communication. Okay, so the first thing we have is our terminal bouton here. This is your presynaptic neuron. And then we have our second neuron. Okay, this is our postsynaptic neuron. Okay, and this region here, this space in between these two neurons is your synaptic cleft. Now, let's put our structures in. So we said that we have these vesicles. Right, we got a few vesicles here. And these have neurotransmitter in them. Okay. We also have the presence of a voltage-gated calcium channel on this membrane, which is allowing the influx of calcium. And then on the adjacent membrane, we have a receptor here. Right. We have this little enzyme that's degrading any excess neurotransmitter. And then we have this reuptake molecule here. Okay. So the first thing we have is an action potential traveling down the length of an axon, and it's going to get to, this is your action potential, it's going to get to the terminal Butan. And normally at rest, we know that a neuron is positively or negatively charged inside. So at rest, is a neuron positively or negatively charged on the inside? Anyone? Negatively charged. So we start out at rest with this neuron being negatively charged in here. When the action potential gets to the terminal Butan, that's the first step here, the inside of this cell now becomes more positive. Okay. The second step is these voltage-gated calcium channels, so I'll write here voltage-gated calcium channel. These are going to open up and they're going to allow the influx of calcium. Okay, so our first step is the action potential arrives at the axon terminal. The second step is that this opens up voltage-gated calcium channels, which are sensitive to the membrane potential at the terminal Butan. Calcium rushes into this terminal Butan, so we got calcium rushing in. And that triggers a process called exocytosis, where this vesicle approaches the membrane, and the membrane of the vesicle fuses with the membrane of the terminal Butan, and all of this neurotransmitter is released out. Okay, so we have the release of this neurotransmitter into the synaptic cleft. Our fourth process here, or our fourth step, is the binding of this neurotransmitter to some type of receptor. So this is your receptor here. And then after we bind the neurotransmitter to this receptor, we're going to bring about some type of cellular response. Okay? Now, I don't want to confuse you, so we're not going to talk about exactly the mechanism here. We'll come back to this, but binding of the receptor to, binding of the neurotransmitter to the receptor is what eventually brings about the response on this cell, okay? And that can be different depending on different things. So we'll talk about that in a bit. The next thing that happens is if there is excess neurotransmitter at the cleft, it will then be degraded by this enzyme. So this enzyme degrades excess neurotransmitter. Okay. And then after we've degraded these two neurotransmitters, or after we've degraded the neurotransmitters, those degraded parts can then be reuptaken back into the terminal Butan, and they will be repackaged into new vesicles that can be recycled. Alrighty, and this is how communication takes place at the chemical synapse. Right, you have your presynaptic neuron, your postsynaptic neuron. The action potential arrives at the terminal Butan. This triggers the opening of voltage-gated calcium channels. Calcium rushes into this axon terminal or terminal Butan. That then triggers exocytosis, where this neurotransmitter is released into the cleft. This neurotransmitter will diffuse across the cleft and bind onto this receptor. And again, the receptor and the neurotransmitter are unique to each other. Okay? That will bring about some type of cellular response depending on the location of this receptor and the type of neurotransmitter and so on and so forth. To cease this signal, this enzyme will degrade any excess neurotransmitter at the cleft, and those degraded pieces will be reuptaken back into the terminal bouton, repackaged, and recycled to make new neurotransmitter vesicles. Okay? Alrighty, any questions? Any questions on that? Okay. Is everyone done with this? Yes? All right, so let's continue. Okay, so these are the steps we pretty much just drew, and you want to spend some time trying to commit this to your memory. This particular sequence of events will not go away. We're going to see this at the neuromuscular junction. We're going to see this at the neuroeffector junction. We're going to see this in cardiac myocytes. So practice drawing out this sequence of events. Practice listing the steps of what's taking place, and make sure you really understand the basics of this. Okay? Because we're going to build on that. Now, there's an idea called the synaptic delay, and the synaptic delay is basically a pause or a 0.5 to 5 millisecond delay between the arrival of the action potential and the change in the membrane potential on the opposite side, or on the postsynaptic side, rather. So I'll say that again. There is a slight delay of about 0.5 to 5 milliseconds between an action potential getting to the terminal bouton and the action potential actually influencing another action potential on the postsynaptic side. This is called a synaptic delay. And the main reason for the synaptic delay is the exocytosis, or the release of the neurotransmitter, which is caused by the change in the calcium channels. Okay, so it's not having to do with the time it takes for this neurotransmitter to diffuse. Okay, and that can be a misleading idea. So it has nothing to do with the time for diffusion of this neurotransmitter. What is causing this delay is the exocytosis, the time it takes for these vesicles to get to the terminal bouton, to fuse with the membrane of the terminal bouton, and to exocytose, or to release those neurotransmitters. Here, this is your synaptic delay. Synaptic delay.